in the classes only, and it will be very much convenient for you, right? Now coming to the second thing which we have is we give personalized attention to each and every student. अभी कैसे ये हो सकता है? Because in each of our batches maximum 6 students रहते हैं. This can be like one student batch or two student batch, as per you will choose, and definitely maximum to maximum 6 students रहते हैं. This will help you to ask and clear all your doubts in the classes. There will be no miscommunication, as well as the teacher can also keep an eye on you, so that how you are progressing, where you are lagging, everything will be marked and you will get the feedback, and this feedback will help you to improve, right? Now coming to the third thing which we have is a very good dedicated academic mentor team who continuously track आप कैसे कर रहे हो homework, आप सारे homework ठीक ठाक से कर रहे हो या नहीं, सारे class work में आपका ध्यान कैसे है, and definitely in which topic you were already prepared, which topic you were lagging. So definitely we will focus on those topic where you were lagging, and you will prepare each and every topic in a very nice way. Okay, and the fourth thing which is very important, that is we cover the syllabus as per your school examination schedule. So if you have any topic which is coming in your school examination, you can tell it to us, we will do those topic, and then we will move on to the rest of the topic. So this type of custom request generally we handle. So these are basically the general features of Swiflearn, and let us now see why this Swiflearn is different from any other platform. First of all, we maintain a detailed progress report of each and every student. And जैसे आपके स्कूल में भी PTM होता है, उधर progress report आपको मिलते हैं, तो इधर भी same to same होता है. We will have the PTM. Your parents will meet with us directly. We will converse, and they definitely if they have any feedback, they will give it to me, and we will also give the feedback, so that mutually ये होगा कि आपका total overall development बहुत अच्छे से होगा. Okay, so this progress report will help you to grow properly. Secondly, we have the SAT, that is the Swiflearn Academic Test Series, which will help you to know what is your rank all over India, and it will happen in every Sunday from 9 a.m. to 5 p.m. Right? So anytime you can perform over here, and your rank and name will be given on the leaderboard. Fine. Coming to the very important, that is the practice. You already know that practice makes a man perfect. So after completing each and every topic, what you have to do, you have to practice the question answers, right? So question answers may be objective or subjective, and definitely we have the unlimited practice test. So you, after completing each and every topic of whatever subject it is, you can go for the practice test, do practice the question answers, okay? And mostly you lose the marks in the subjective question. Yes. Hi, Cracker, what is your name, beta? Please tell me your name in the chat box. We are basically doing the germination today, okay? Yes, seed germination. Acha, so what I was telling is basically you lose the marks in your subjective question. 3 mark या 5 mark का question आते हैं, and you sometimes miss out क्या लिखना है, what are the key points to include in your answer, right? And that is why you do not get the full marks. So here when you will practice writing the subjective answer, there is a group of teacher, definitely they will check your answer, and you will be able to understand where to include the diagram, where to include the chemical equation. It is totally dependent upon the different subjects. Okay, so this will happen, and this will definitely help you to understand what is your progress and how you are improving. Okay, so to experience all this thing, what you have to do is you have to book the free trial class. Trial classes are all free, and then enroll your name in our regular batches. So regular batches में हम रोज मिलेंगे, and we will see we will discuss a lot of different topics, right? Yes. Chalo. So let us now see what we will study, and before that let me remind you that to subscribe our channel and press the bell icon, so that you will never miss any of the update of the upcoming session, and do not forget to like, comment and share also. Okay, chalo, let's move on to the seed. So we have already done 2 sessions of seed, which is there in ICSE board syllabus of class 9th science. So seed क्या होता है? What are the different types of seed? उसका structure क्या है? Dicot and monocotyledonous seed का structure हमने already देख लिया in session 1 and session 2 ने हमने already देख लिया कि what is germination and germination क्या है? What are the different types? Like कैसे germination होता है? What are the different types of germination, like the hypogeal and the apigeal germination, right? So now let us focus on the experiments which are determining the role of different factors in the seed germination, कि क्या क्या different external and internal factor हो सकते हैं जहां seed germination को जो help करते हैं. Okay, and then we will move on to the germination in some common seeds. Fine. Yes. Now, can you tell me, in the last session we have already studied about the germination. तो germination क्या है? Can anybody tell me? Yes. Can anybody tell me what is germination? And also, can you please tell me कि what are the external factors which help in the germination of the seed? And as you already know that whoever will give me most of the answers correctly, he or she will be the student of the session and I will write your name on the screen. ठीक है? Yes? चलो. So, now let's move on to the condition of the seed germination. We already have an idea कि the different conditions which are needed for the seed germination is specifically the proper temperature, amount of water and the air, right? So airs में से mainly oxygen will help them to grow and germinate, right? So this oxygen which is coming from the air, water and a proper suitable temperature, which is very much important for the seed germination, all of them need it. It is needed by the seeds. Okay, now let us discuss with the help of the experiment ki without that what will happen, without all these factors what will happen to the plant and why they will not be able to do the germination part. First of all, the 3 bean seed experiment हम देखेंगे. तो देखो इधर क्या है experiment setup में, you have to take a beaker of water. Now within the beaker of water you have to take one glass slide. ठीक है, अभी इस ग्लास साइड में टेक 3 bean seeds and attach it with the glass slide. तो ऐसे करके अटैच करोगे कि one seed will be totally immersed in water like this, you can see. Now second one will be half immersed in water, मतलब वो थोड़ा सा water में है और थोड़ा portion उसका air में है, so that it can get both the air and the water and also a proper temperature, right? And next is another seed where which will be in contact with the air only and no water, right? So what will happen if we keep it for some days? Yes, Anushka, can you tell me what will happen to the seeds if we keep it for some days? Yes. Any idea? We will basically see that the germination will take place only to the middle seeds. Why? Because the seed which is in between the air and water, it is getting a proper amount of water, then it is getting a proper amount of air, as well as it is getting a proper amount of temperature, a proper optimum temperature, and this will help the seed to germinate properly, right? So this is the observation that the seed which is partially in water and in air, that seed will germinate, the middle one, and the seed which is directly in contact with the air but no water, definitely it will not be able to germinate. Very good, Anushka. Fungus will come if exposed to air but no water. And the seed which is totally immersed in water, it is not basically getting any oxygen, right? So dissolved oxygen jitna bhi hai, so it will be like it will be taken by it and there will be no available dissolved oxygen, and that is why it will not be able to germinate. Fine, up to this much clear hai? Yes, so here we can see that the water is very much important for the germination, as well as the air, as well as the proper temperature. Now we will focus one by one to each of them. Shaurya doesn't really matter, beta. Germination will also be there. It is a bio topic, so if you were in CBSE, that is also not at all a problem. Okay, you will get some more information from his. Chalo. So let's do another experiment on germination to see that water is very much important for the seed germination. So over here you have to take two seeds. Some of the pea seeds you have to take it in the dry cotton wool, and some of the pea seeds you have to take it in the moist cotton wool and keep it for 2 to 3 days. Where will you see that germination will happen? Definitely in the beaker too, where there will be the moist cotton wool, because if you keep the seeds in the moist cotton wool, it will get the air, it will get the water from the cotton, as well as it will get the proper temperature, and that is why the gram seeds will grow where the cotton wool is moist. But if you just keep it inside the dry cotton wool, then you will not be able to see that germination will happen, because there is no supply of water, right? And that is why we will be able to prove that water what is very much important for the germination of the seed. Okay. Now you may ask the question like, why this water is so much important for the germination? So the answer will be what? Definitely the food which is stored within the seed, that is totally the dry food, and the embryo will not be able to take that dry food. So the food to dissolve within the water, which will be basically taken by the embryo, the food should be in a liquid form. Okay, as well as the food should get a proper dissolved oxygen also, right? Yes. So excess water, जैसे without the water the seed will not be able to germinate, as well as the excess water भी इसको stop कर देंगे germination से. क्यों? Because whatever oxygen is already dissolved within the water, it will be totally taken up, right? And this germination will not possible if there is no proper amount of oxygen within the water. Now oxygen is lacking, and that is why fully if it is immersed inside the water, then also the seed will not be able to germinate. Yes. Yes, Anushka, your answer is totally correct, that when the seeds are kept in water or the seeds are provided with moisture, yes, it will imbibe and the micropyle will basically absorb the water, right? We have already studied it in the previous session, and then it will help the seed to swell and the seed coat will burst and germination will take place. Take care. Now let's move on to the next type of experiment, that is where we have to prove that air is also important for the germination of the seed. अभी देखो क्या करना है हमें. Take some seeds of pea or gram and place it on the wet cotton wool in a beaker A. Similarly, some water is boiled in beaker B, so that जो भी dissolved air है boiling water में, मतलब water में, that we have to remove. Okay, after removing the water, after removing the excess air or the oxygen bubbles from the water, you have to put some oil in it. Okay, so that even if the water come in contact with the air, there will be no exchange of the air molecules. Fine. So what will happen in that case? In which of the following cases the seed germination will take place? Definitely in the first one. Why? Because in the first one you can see that it is coming in contact with the air, so it is getting the oxygen. It is also getting a proper temperature, as well as we are using the wet cotton wool, it is also getting the water. So air, water and proper temperature, ये तीन चीजें already it is getting, and that is why germination is favorable for here. But in this case where we have taken excess of water, which is the boiled water, and boiled water में air नहीं है, because we have already removed the air, and also it is not in contact with the atmosphere because we have dropped some of the oil. So over here the seed will not be able to germinate, as there is total absence of air. Got it? Yes, so here also we can prove that air is very much important for the germination of the seed. Without the air, seed will not be able to germinate because cell division will not take place. And cell division अगर बंद हो जाएगा तो ऑक्सीडेशन भी बंद हो जाएंगे, उसके अंदर जो रेस्पिरेशन, मेटाबॉलिक, फिजियोलॉजिकल फंक्शंस या फिर मेटाबॉलिक एक्टिविटी स्टार्ट होता है, ये भी सब बंद हो जाएगा एंड टोटली द सीड विल नॉट बी एबल टू germinate. So when it is getting the air, it is also get helping it for the cell division से उसको energy मिल रहा है for the respiration, and this respiration will help them to grow in a proper way. Fine, up to this much totally clear है? Yes Anushka. Chalo, now let's move on to the third factor where we will see again seed germination is very much important with the help of a proper temperature. So हमने already prove कर दिया कि water चाहिए, air चाहिए, नहीं तो properly germination नहीं होगा. And third factor हम अभी देखेंगे कि कैसे temperature भी important होता है for the germination of the seed. Fine. Over here, you have to take some gram or P and keep it in the two beakers, beaker A and B, on a wet cotton wool. Right? अभी आप क्या करोगे? एक beaker को लेंगे, can be the beaker A, and place it in the ice box. तो आइस बॉक्स रेफ्रिजरेटर में मिल जाएगा और उधर आप प्लेस कर दो. एंड यू कैन सी आफ्टर सम टाइम व्हाट विल हैपन. ठीक है, what will happen? What do you think? 2-3 days के बाद आपको क्या मिलेगा? बीकर ए में जर्मिनेशन होगा या फिर बीकर बी में जर्मिनेशन होगा? यस, बीकर ए या बीकर बी? Definitely beaker B में germination होगा because beaker A you have kept inside the refrigerator in the ice box, right? And it is not at all getting a proper temperature. So definitely germination will not take place where it is not getting a proper temperature. तो इसी में germination नहीं होगा. But when you are taking the keeping the seeds inside the cotton wool in the beaker A, germination will take place properly. Very good Anushka. Yes, beaker B में germination नहीं हो, beaker B में germination होगा, and beaker A which you have kept inside the ice box, germination उधर नहीं होगा due to not getting a proper temperature over here. अभी ये proper temperature का मतलब क्या temperature चाहिए? Usually optimum temperature which will be very much favorable for the germination process is 25 to 30 degrees centigrade. Always you have to keep it in mind. 25 to 30 degrees centigrade में जब temperature होता है तो सबसे ज़्यादा high speed में germination हो सकता है. If it is below 25 मतलब आप सोच लो कि 0 to 10 degree में बहुत slowly germination होता है, पर होता है. Below 0 degree centigrade, no germination will be happening, and above 50 degree centigrade also germination नहीं होगा क्योंकि germination अभी 30 to 35 या फिर 35 से 40 तक अब जब जाएंगे तो टेम्परेचर जब थोड़ा थोड़ा धीरे धीरे बढ़ जाएंगे तो जर्मिनेशन का रेट भी स्लोली इट विल बी रिड्यूस्ड. ओके, जर्मिनेशन का रेट भी कम हो जाते हैं. Okay, and when it will reach or it will cross 50 degree, totally germination of the seed will be stopped and germination will not happen. We will not get the baby plant. ठीक है, तो suitable temperature is very much important for the activity of the embryo. embryo के अंदर जितना भी physiological activity होता है which will be like totally destroyed when it is kept in the high temperature, and it will be totally stopped if you or dried up if you keep it in the very low temperature. So optimum temperature for the growth and development of the seed will be 25 to 30 degrees centigrade. It is very much important for the growth, right? Beaker is a vessel, beta Anya. It's a vessel where you can do the different types of experiments. Okay, now Let us now see some of the germination in some common seeds. So common seeds matlab you have already, you can do it at your home also. These experiments are very easy experiments. You can do it at home only. Utna highly complicated chemicals bhi nahi chahiye. What you only have to do is you have to take some water, cotton wool, some seeds and do the experiments. ठीक है, तो करके देखना है ये टेम्परेचर वाला, एयर वाला और वाटर वाला एक्सपेरिमेंट which we have already discussed in today's session. And now we will see some of the common seeds कैसे germinate करते हैं. तो common seeds मतलब you can take some of the castor seed, you can take some bean seed, maize seed, ये आप ले सकते हैं and you can do this experiment. Okay. Yes, Anushka, very good, very nicely explained. Beaker is a glass container with flat bottom. Okay, flat bottom भी होता है, beaker का round bottom vessels भी होता है. Okay, चलो, तो mostly it is the flat bottom. You were totally correct. ठीक है. Now, first we will see the bean seed germination. So, what you have to do, you have to take the bean seed, keep it inside the soil. The soil must have a proper amount of oxygen also, because the soil have to be very much loosened so that the seed will get the proper amount of oxygen, water and a proper temperature. Fine. Now, when the seed will get a proper favorable condition, what will happen? The seed coat will swell up due to the imbibition and the embryo will emerge out by rupturing of the seed coat. Fine. तो rupturing of the seed coat में कौन first emerge out होगा? Radical. है ना? तो radical will come out and it will go deep inside the soil because it is forming the root system. We have already studied it in the previous session. है ना? So radical will give rise to the root system and the plumule will give rise to the shoot system. Fine. Now this hypocotyle which is present over here will grow and it will become straight. Yes. तो straight होने के लिए ये क्या करेंगे? It will pull the cotyledon towards the above of the ground. So what type of germination it is showing? Yes. We have already studied about the apigeal and the hypogeal germination. So what type of germination is shown by this bean seed where the cotyledon is pulled above the ground? Yes. Quickly tell me. It is the apigeal type of germination, right? So what will happen? The cotyledons with the green leaves will come up and उससे पहले जो उसको खाना देते हैं या फिर nutrients देते थे, तो it would get the nutrient from the endosperm. Later on, it will get the nutrient from the trees. Sorry, the leaves. The leaves will show the photosynthesis and finally it will provide the nutrition to it. ठीक है, तो दिस इज नोन एज द एपीजियल जर्मिनेशन. तो हम और एक एपीजियल जर्मिनेशन का एक्सपेरिमेंट देख लेते हैं. दिस इज द विद द हेल्प ऑफ द कैस्टर सीड. ठीक है. नाउ कैस्टर सीड जर्मिनेशन भी सिमिलरली होता है एंड इट विल टेक लाइक अराउंड अबाउट 7 days, 1 week में हो जाते हैं. So how will it take place over here? See, there is a caruncle. What do you mean by caruncle? It is nothing but a membrane-like structure which is present above the micropyle. तो micropyle का काम क्या है? Absorption of the water, right? So when it will absorb the water, the caruncle will go out or it will just give them the proper space, and from there the radical will go out. The radical, when it will grow out, it will go down towards the soil. It will try to absorb more and more water. And the embryo, the hypocotyle will again enlarge, become straight. And it will again move the cotyledon above the soil. Yes, very good, hypocotyle. So the type of germination is known as apigeal germination, right? So after the formation of the leaves, what will happen? The cotyledons will fall off and independently वो germinate हो जाएगा, and this type of germination is known as the apigeal germination. ठीक है? Yes, so castor seed, bean seed, these are the common seeds where we can see the apigeal type of germination. तो इधर हम सब देख रहे हैं कि the three main factors which are very much important for this type of germination is the water, air and a proper temperature. ठीक है? These are the three main external conditions. Coming to another type of germination where we can see the maize grain germination. तो maize grain कैसे germinate होता है? First of all, you have to put the seed inside the soil, having a proper supply of air, water and temperature. Then the grain will absorb the water and it will swell up. ठीक है? अब देख रहे हो maize इधर है, the grain will absorb the water, and after absorbing the water, what will happen? You already know that this is a monocotyledonous seed, right? So monocotyledonous seed का दो portion we have already studied. Remember the structure of the maize seed. It has the endosperm and it has the embryo. So embryo में one portion is the coleoptile, another portion is the coleorhiza. Remember? So this coleoptile will grow and give rise to the plumule. The sheath which is covering the plumule is the coleoptile. So it will grow and give rise to the plumule, which will again grow upward to the soil and give rise to the shoot system. And lower portion में रहता है coleorhiza, which is basically guarding the radicle, and the radicle will grow downwards towards the soil and it will give rise to the root system. Okay, so this is how the germination of the maize grain will take place. There the seed will remain inside the soil and it will not come up. Okay, and that is why this type of germination is known as the hypogeal germination. Hypogeal germination मतलब वो नीचे ही रह जाएंगे, and whenever totally the seed will be exhausted with their nutrient content and everything, it will dry up and it will just mix with the soil. ओके, वेरी सिंपल तरीके से ये मेज ग्रेन का जर्मिनेशन होता है. अंडरस्टूड? अप टू दिस मच क्लियर? Yes, it's a very simple thing which we are studying today because we have mostly completed with the structure of the seed, the different types of germination, the different types of seed in the previous two sessions. If you were absent in the two previous sessions, please go with those sessions also. Videos will be available in our YouTube channel. And then try to go with this session, that is the experiments and all. This will help you to understand. Fine? Yes. So this is all about the maize grain germination. कैसे germination होता है. So this is all about the chapter also. Let us now discuss quickly the question answers. So first of all, the first question, these are the questions which will come for your like subjective answers. Okay. So 3 mark, 5 mark का question ऐसे होगा. So let us discuss this type of question and then we will move on to the objective types. ठीक है? Chalo. My first question is the description of the structure of the castor bean seed. You have to explain the structure of the bean seed. As you already know the structure of the bean seed, it is the dicotyledonous seed. You have to give a proper explanation of the structure. जैसे the seed coat, it is covered with a seed coat. Seed coat का दो layers होता है, testa and tegment. And along with that, please try, if you are getting this question with like the 5 marks, in 5 marks question, then try to give the diagram also. Okay, so the scar which is the hilum, उसके ऊपर रहता है micropyle which will help, a very small pore which will help the seed to absorb the water. Okay, yes, very good. Kalinka, which is basically a type of elliosome. Exactly, Anushka. Okay, so hilum, micropyle, this you have to like explain. Then you have to explain the embryonal axis and two cotyledons. Cotyledons are basically fleshy. They generally reserve the food for the developing embryo. And you have to mention the radicle and the plumule, which will give rise to the root system as well as the shoot system of the plant. Okay, so the whole complete answer will help you to get the full 5 marks. Along with the diagram, you have to give the proper labeling. So don't forget to give the diagram and labeling if you were getting the same question as 5 marks. Okay, my second question was the explanation of the fruit and the grain. What do you mean by fruit and grain? Fruit, you already know that it is nothing but the ripened ovary. The ovary, after fertilization and the growth of the embryo, the total ovary of the flower will be converted into the fruit, and the ovules which are present within the ovary will be converted into the seed. We already know that. And the fruit is basically including the seed. Yes, तो हम क्या करते हैं, फ्रूट्स को खा लेते हैं और सीड्स को डिस्पर्स कर देते हैं, so that within the seeds the embryo are growing, and after the proper after getting the proper condition, it will be able to grow into a new plant. Yes, this is nothing but a very simple type of sexual reproduction which we generally see in the flowering plants, right? Now grain क्या होता है? Basically, grain is nothing but one-seeded fruit where the fruit wall and the seed coat are fused with each other to form the proper protective layer. So this type of fruit is known as the grain. Okay? Yes? Okay. With or any other cultivated? Yes, very good, Anushka. Very nice. Yes. Now coming to the third question. My third question was, what were the main role of the micropyle in our seed? In the seeds, micropyle का role क्या है, function क्या है? इसका main two functions होता है. First and very important primary function तो होता है to absorb the water. Yes, the water is very much important for the growth and development of the embryo, so they need to absorb the water, and this absorption of water is done by the micropyle. It's a pore which is present just above the hilum, right? And the second function is nothing but the diffusion of the respiratory gases. That there is a respiratory gases, definitely the oxygen, which will be also needed for the embryo for its growth and development. तो ये diffuse होके भी embryo के अंदर आते हैं due to the presence of the micropyle. So these are the basic two functions which are played by the micropyle in the growing seed. ठीक है? Yes. Okay. So this is all, and definitely the cotyledon will provide the nutrition to the embryo. Now let us move on to quickly the ऑब्जेक्टिव क्वेश्चन. अभी मैंने तो ऑलरेडी सब्जेक्टिव क्वेश्चन या फिर लॉन्ग आंसर वाला क्वेश्चन मैंने ऑलरेडी डिस्कस कर दिया उसका आंसर, क्योंकि इट इज़ नॉट पॉसिबल फॉर यू टू राइट इट इन एवरीथिंग विद द एक्सप्लेनेशन इन द चैट बॉक्स. तो मैंने ऑलरेडी हाउ टू राइट द आंसर अकॉर्डिंग टू द मार्क्स, लाइक 2 मार्क्स में, 3 मार्क्स में, 5 मार्क्स में कैसे लिखना है वो मैंने बोल दिया. Now you quickly tell me the answer of this MCQs. Okay. So first question is, the food is stored in the seeds of castor. Where is the food stored? Is it in the radicle or plumule or in the cotyledons or in the endosperm? Where is the food stored in case of the castor seed? Quickly tell me the answer. Yes. Let me see. Quickly tell the answer. अनुष्का, will you be able to see? Will you be able to say कि castor seed का food कहाँ store होता है? Radicle में, plumule में, या फिर cotyledons में, या फिर endosperm में? It will be stored in the endosperm, right? Yes, the food is always stored in the endosperm, which will give the proper nutrients to the growing embryo. Always remember. ठीक है? Coming to the next question, the caruncle, which is present in the it is present in which type of seed? ये wheat में रहता है या maize में रहता है या फिर paddy seed में paddy में रहता है या फिर castor bean seed में रहते हैं? Yes, quickly tell me the answer. Caruncle is present in which seed? I've already mentioned that it is a membrane-like structure which is present at the opening of the micropyle, right? Yes, so this is present in which seed? Okay, Anushka, I'm waiting for you. 1 minute. Okay. Tell me in which seed we can find this caruncle. As I can see in the chat box that you have already given me this answer, right? Previously, you have already mentioned that where we are getting this caruncle, we basically get it in the castor bean seed. Yes, in the castor seed, we can get this caruncle. Now, let's move on to the next question. An essential condition for the germination. What is the essential condition for the germination? You can see already this answer is given. That is water, temperature and oxygen. All these three are very much important for the germination of the seed. And hypogeal germination इसमें से कौन से seed में होता है? As we already know, apigeal germination takes place in the pea, bean and castor seed, where the seed will go, the cotyledon will go above the ground during the germination. But only hypogeal germination we can find in the maize, right? We have just done the germination of the maize seed. The cotyledon will remain inside the soil when the radicle and the plumule is coming out, right? Yes. Chalo. Next is the largest seed. Which one is the largest seed? You can see that. Please note it down and write it in your copy also. This type of question you may get. That is the double coconut will be the largest seed. Okay. And collective terms for the stages where the formation of the seedling will take place from the seed. It is nothing but the germination. Dormancy is not, because we have already studied about the dormancy. It is a condition of the seed where there will be no germination. Even if it is getting a proper condition for the like temperature, air, water and everything, then also the germination will not take place properly, and that condition of the seed is known as the dormant seed. Fine. And viability will also not be the answer. So germination is the process where we can see that the seedling is growing from the seed. Yes. Now let me give you one fact that now the research is going on about the germination of the seed in various laboratories in all over the world. So Moser, he is a professor of horticulture and landscape architecture, and Kenzie Mura, the former Purdue postdoctoral researcher, now is the assistant professor of Tsukuba University in Japan. They discovered that the step involved in keeping the seeds from germination in adverse condition, बहुत adverse condition जैसे drought condition बोलो या फिर बहुत freezing very low temperature में कैसे seed germination होता है, इसके ऊपर अभी ये research चल रहा है. Fine? Yes. So let us now see what we have already studied. We have discussed about mainly the experiments. आज का सेशन में उतना ज़्यादा important, मतलब important तो है, उतना ज़्यादा complicated कुछ नहीं था. तो हमने already discuss कर लिया कि 3-bean seed experiment क्या है and why this air, water and the temperature is needed for the seed germination, without which what will happen, germination will not take place. We have studied this, and we have studied about the seed germination of beans, castor and maize, right? So this we have already completed in the today session. Okay, so thank you very much for being with us till the end of the session. And today's student of the session is Anushka. Okay, so Anushka is the student of the session. So clap for her, everyone, and thank you for being with us till the end and giving me all the answers correctly. And definitely we will again meet in the next session. Before going, let me again remind you that please subscribe our channel and press the bell icon so that you will never miss any of the update of the upcoming session. We will meet in the next session again with a new topic, and don't forget to like, comment and share also. Okay, so let it be up to this much for today. We will again meet in the next session. So till then, bye bye, take care, stay safe and stay healthy. Bye bye.